The phenomenon of perceiving colours that are undetectable by dogs can be attributed to the scientific explanation of lab-grown retinas. Greetings and salutations to everyone. In today's video, we will be delving into the fascinating research conducted by scientists at Johns Hopkins University. Their groundbreaking work involves cultivating human retinas in a laboratory dish, allowing them to explore the intricate mechanisms behind colour vision. Unlike dogs, cats, and other mammals, humans possess the remarkable ability to perceive a vast array of colours thanks to a derivative of vitamin A. The findings, recently published in PLOS Biology, not only shed light on conditions such as colour blindness and age-related vision loss, but also challenged the previously held belief that thyroid hormones controlled the development of colour-sensing cells in the human retina. So, without any further delay, let's dive into our discussion. Researchers made a groundbreaking discovery by growing human retinas in a petri dish, revealing the role of a derivative of vitamin A in the development of specialized cells responsible for human beings' unique ability to perceive millions of colors, a capability not shared by other mammals like dogs and cats. Author Robert Johnston, an associate professor of biology, expressed that the utilization of retinal organoids provided an unprecedented opportunity to examine this distinctly human characteristic. Exploring the factors that distinguish us as humans is a significant inquiry. In a groundbreaking study published in PLOS Biology, new discoveries have emerged that contribute to our knowledge of color blindness, age-related vision decline, and various conditions associated with photoreceptor cells. These findings shed light on the intricate mechanism through which genes direct the human retina to produce specialized color-detecting cells. This significant breakthrough challenges the previous belief that this process was solely regulated by thyroid hormones. Through the manipulation of the cellular characteristics of the organoids, the team of researchers made a significant discovery regarding the role of retinoic acid in determining the specialization of cones in perceiving either red or green light. It is worth noting that only humans with typical vision and primates that are closely related possess the ability to develop the red sensor. For many years, scientists believed that red cones in the eye were formed randomly, as if through a coin toss, where cells would randomly choose to sense either green or red wavelengths. Recent research from Johnston's team hinted at the possibility that thyroid hormone levels might play a role in this process. However, the latest findings propose a different explanation. Red cones actually develop through a precise sequence of events guided by retinoic acid within the eye. During the initial stages of organoid development, the research team discovered a strong association between elevated retinoic acid levels and an increased abundance of green cones. Conversely, a decrease in retinoic acid levels altered the genetic instructions of the retina, resulting in the production of red cones during later stages of development. According to Johnston, while there may be a degree of unpredictability involved, the key discovery is that the production of retinoic acid occurs during the early stages of development. This precise timing holds significant importance in unraveling the process of creating cone cells and its implications for learning and comprehension. The cone cells responsible for detecting colors in the human eye, specifically the green and red varieties, share a striking resemblance. However, it is the presence of a light-detecting protein called opsin that distinguishes these two types of cones and informs the brain about the colors perceived. The determination of whether a cone becomes a green or red sensor is dictated by different variations of opsins, even though the genetic makeup of each sensor remains remarkably similar, with a 96% similarity. Through the utilization of a groundbreaking technique capable of identifying these subtle genetic distinctions in organoids, the research team was able to observe changes in the ratio of cones over a period of 200 days. Author Sarah Hadiniak, who conducted the study while pursuing her doctorate in Johnston's lab and is currently affiliated with Duke University, explained that by manipulating the composition of green and red cells in organoids, they are able to influence the predominance of either color. This manipulation holds significant implications for understanding the precise mechanisms through which retinoic acid influences gene expression. One of the most unexpected discoveries of the recent study was the observation of how the ratios of green and red cones in the retinas of 700 adults differed significantly. The researchers meticulously mapped these variations in cell proportions, as noted by Retinas. The variation in the ratio of green and red cones without any impact on visual perception continues to baffle scientists. According to Johnston, if the same kind of cellular variation determined the length of a person's arm, the resulting arm lengths would be remarkably diverse. In order to enhance comprehension of conditions such as macular degeneration, which leads to the deterioration of light-sensing cells in the central area of the retina, the researchers are collaborating with various laboratories at Johns Hopkins. Their objective is to further their knowledge regarding the connection between cones, other cells, and the nervous system. According to Johnston, the ultimate aspiration is to provide assistance to individuals experiencing vision impairments. Although it may take some time before this becomes a reality, the fact that we are capable of generating various types of cells is incredibly encouraging. Among the authors affiliated with Johns Hopkins University, notable names include Chiara C. Eldred, Boris Brenneman, Katarzyna A. Hussey, Joanna F.D. Hagen, Rajiv C. McCoy, Michael E.G. Soria, and James Taylor. Additionally, James A. Kuchenbecker, Thomas Ray, Ian Glass, Maureen Knights, and Jay Knights from the University of Washington contribute to the academic community. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.